Okay, are there any questions before we begin? I guess I should ask, can somebody confirm you can hear me?
I can hear you.
Great. My computer is going to be working slow while it's still making the Zoom file for the lecture, so please bear with me. Am I sharing my screen?
No. I
All right, I'm not. Okay. I clicked on the button to share and nothing happened, and I did it there. So, all right, we're 97%, so we're almost done. All right, good. All right, today we're going to talk about Lab 9, identification of gram-negative organisms. And let me state that I have graded some of the earlier labs. Let me stop sharing the screen for just a minute. Oh, good. That means the lecture is now finished. And let me take a look at the grade book and then I can tell you which labs I've graded. I think it's through Lab 4 has now been graded 2 times. I'll close down. And then Lab 8 was graded 1 time. You can make corrections for Lab 8 up until this Saturday. Let me look on that. You've got labs all the way from lab 0, 0 to lab 4 graded 2 times now. And it looks like I didn't get one person graded on lab 4. All right. And lab 8 has been graded and you can make corrections until this Saturday. There's another worksheet in the lecture. I meant to mention that in the lecture that almost everybody did wrong. Actually, the first student did it right, and there may be 2 other students in the class who did it right. And on worksheet lecture 6, I think it is. Let me open this up to make sure that's the one. You were supposed to answer all of the questions to each number. So if it was like 1A and 1B, you were supposed to answer 1A and 1B. There was an exception for question 10. You can read about the exception, but most people only answered one question of 1A or 1B, and that wasn't correct. You're going to get marked down if you don't go in and answer another question. Now, you have to answer a question that has not been answered. So if you answer a question that has been answered, you'll get marked down because you're just answering a question somebody else has answered. So you need to go find a question someone else has not answered, and hopefully it's been going in the correct order, and then it'll be easy to figure out what you need to answer and what you don't. And this doesn't look correct because a whole bunch of people are answering question 10, unless it's 10A, B, or C, and they're not putting in the number, the letter. But um, you need to go in there and answer another question. You were supposed to do all of them, like if question 2 had 2A, B, and C, you were supposed to do 2A, 2B, 2C. I'm not going to require you to answer 2 additional questions, but you do have to answer at least one more question. And it has to be an unanswered question. And hopefully you can figure out what students have answered and what they have not. If you go in and do it the correct order, meaning the correct order of the worksheet, it's pretty easy. And initially it looks pretty good, but later on, people were putting in a whole bunch of tens, and I don't know if that's correct. I'll have to go in and take a look. Okay, any question about any of that? Let me get out of the grade book. I can share my screen. And let's go to the syllabus. We're going to be talking about Lab 9, identification of gram-negative organisms. You should still be working on your unknown work. And if you don't get started on that immediately, you will probably not be able to finish the unknown project in time because it will take about 2 weeks to get it finished. Um, on average. Some people will take longer, some people will take shorter. All right, let's go to lesson worksheet 9. There we go. Lab 9. Can you guys see this? Should be able to. Identification of gram-negative bacilli. This is the last lab for the course. Oops. As always, there's some reading. This lab has very little, only a page on biochemical tests and bacterial identification, and a few pages on methods for classifying and identifying microorganisms. There are some video clips as supplemental material in this lab. The learning objectives: upon successful completion of this lab, you should, 1, be able to interpret the results of the following biochemical tests. The Imvik reactions. Imvik stands for indole, I, methyl red, MV, I is added, and then C for citrate. So the Imvik reactions are 1, 2, 3, 4 different tests. These tests are frequently used to classify Enterobacteriaceae, but you can use them for other uh, species as well. We're going to talk about the carbohydrate fermentation test, the motility test, and hydrogen sulfide production. So you should be able to interpret them and give the results for the following tests. And then 2, explain how to interpret the TSI, triple sugar iron slant test, the SEMS, that's sulfur iodine. What's the D stand for? Oh, M, sorry, motility test and the D test. And then understand the growth on McConkey's auger and Hectone and Terrick auger plates. And then 3, be able to use biochemical tests and analysis to solve case studies involving gram-negative bacterial infections. Oops, going too far. So members of the family Enterobacteriaceae are all small gram-negative, non-spore forming bacilli. They are all facultative anaerobes, and there's a lot of species in the family Enterobacteriaceae, including 53 genera. 24 of these genera are known to cause human infections. The members of the family Enterobacteriaceae are all glucose fermenting, and they're all oxidase negative organisms. These are some of the genera of the family, and most of these genera are normal microbiota of the intestinal tract, and that's why they're called coliforms, because they grow in the colon and the intestines. And there are opportunistic pathogens in that they can cause urinary tract infections, wound infections, pneumonia, and septicemia. Septicemia is just blood infections. Common genera responsible for causing opportunistic infections include Escherichia, such as E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several genera of the family Enterobacteriaceae are human pathogens that cause gastroenteritis. These include organisms called in the genera Salmonella, Shigella, and E. coli Suravar 0157H7. So normally E. coli, when it's in your gut, that's it's normally supposed to be, and it's actually a good thing to have in your gut. But Saravar 0157H7 is a human pathogen. This 0157 is an antigen, so is H7, a different antigen. And these antigens make E. coli a human pathogen. Any question about any of that? All right. Pseudomonas aeruginosa is similar organism to the family Enterobacteriaceae, and so all are the members of the Pseudomonaceae family. And that they're all gram-negative bacilli. However, the Pseudomonas does not ferment glucose, so non-glucose fermenting. They're all oxidase positive. They can cause opportunistic pathogens, particularly Pseudomonas aeruginosa, especially in immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa is a healthcare acquired infection, which is also called a nosocomial infection. And you can find it in almost anything that is liquid, hospital drains, sinks, water, even mild soap solutions. It's important for microbiologists to be able to identify and perform antibiotic susceptibility tests on gram-negative bacilli once they're uh, brought to the lab for bacilli cultivation. In the lab, common biochemical tests will be discussed used to identify gram-negative bacilli. And here's a test running a whole bunch of different tests at once. We're not going to talk about that. That's actually running 12 tests at once. We won't talk about that, but we will. Actually, this is in here. We will talk about the indole test. Some organisms produce the enzyme tryptophanase that converts the amino acid tryptophan into the products indole and pyruvic acid. So you have overnight incubation, you add Kovacs reagent, which will react to indole to produce a red color at the surface. E. coli tests positive for indole production. Klebsiella pneumoniae does not. So this doesn't look very red, but it's supposed to be red, and it's positive for the indole test, converting the amino acid tryptophan to indole and pyruvic acid. This is negative for the indole test. When you add Kovacs reagent, it'll be at the top of the liquid, and it's negative if it's yellowish. If it's reddish, and I've never seen it purple like that, but purple, it'll be positive. Any question about the indole test? All right, let's move on to the methyl red and the Voges Prescure test. These are two different tests, the methyl red or MR test and the Voges Prescure or VP test. And I'm not Russian, so I could be butchering the pronunciation of that name. You're looking at the results of the end product of glycolysis with both of the MR and the VP test. The MR test is looking at the production of acid from glycolysis, and the VP test is looking at the production of neutral products from the production of glucose by glycolysis. Both of these tests, if it's positive, meaning acid is produced, the MR test will be red in color, positive. The VP test, if it's positive, it'll also be red in color, meaning a neutral product will be made from glucose glycolysis. Any question about any of that? All right. All right, the MRVP test is initially started in the same tube. So you get one tube and you inoculate it with your test organism, and then you let that tube grow, meaning you incubate it for 2 to 5 days, depending on the test, and then you split that tube into two portions. One portion will be used for the MR test. The second portion will be used for the VP test. Methyl red is added to one portion of the broth. And if the bacteria produce acid, then the pH indicator will remain red, meaning the methyl red is the pH indicator. It will remain red, which is a positive result. If the broth changes to yellow, then it's a negative result. The VP test, you need to take your aliquot of the MRVP tube. So remember, you split it into two. And then you add Barrett's solution A and Barrett's solution B, and you shake it, and then you let it sit. You shake it to add water, oxygen to the tube, and then you let it sit for up to 60 minutes, looking for the production of the red color. You'll get red color if acetone is made. Acetone is a neutral product made from glycolysis. So MR test, positive, it's red, negative, it's yellow. The VP test, positive, it's red, usually in one portion of the tube, such as the top. And then if it's negative, it's the whole tube is yellow. Any question about the MRVP test? If not, let's move on to the citrate utilization test, the last of the IMVIC test. Citrate utilization is used to determine if the bacteria has the ability to use citrate as the sole carbon source in the tube, meaning in the citrate media, the only carbon source comes from citrate. If the bacteria can utilize it, then it can grow on the tube and change the color of the tube to blue. If, on the other hand, it cannot utilize citrate, the bacteria cannot grow, and it will not change the color of the tube, so the color of the tube will remain green. And citrate is a solid media, we call that a slant. The tube will change blue if basic products are made. If basic products are not made or there's no growth on the media, then the tube will remain green. And here we have a positive result. E. coli can metabolize citrate, so it'll be blue. What's that organism which is negative? Don't remember one off the top of my head which is negative. Any question about the citrate utilization test? Oop, I read that wrong. E. coli will be negative in the citrate test. It'll remain green. It does not have the ability to metabolize citrate. Oh, I do know one off the top of my head, which is positive. What the heck is that called? It's not a gram negative, but it's in the unknown project. Micrococcus varians is citrate positive. All right, let's move on to the PR carbohydrate metabolism test. This will be testing the fermentation of a sugar. And if the bacteria can metabolize the sugar, they will make acid end products, which will change the pH of the media. And so the media will change from reddish to yellow. So if acid is made, it'll be yellow. The carbohydrate metabolism test uses one sugar. And so we have glucose, PR tests, so PR glucose, PR lactose, PR sucrose, PR maltose, etc. You just put one sugar in there and then run the test. And so PR carbohydrate is actually a number of different tests, depending on what sugar you're looking at. And you can look at three different results. You can have acid, where the tube turns yellow from the production of acid, and that we have interpretation A for acid. This is negative, interpretation negative, meaning no change or a basic solution. And then you can also have acid and gas, where you have acid being produced, the media turns yellow, and then you have production of gas, which we can see in the inverted Durham tube, that bubble. So AG for acid and gas. If the sugar is fermented, you'll get an acid and you may get a gas. All members of the family Enterobacteriaceae can ferment glucose, so they'll all be at least A. Many of the families, like Citrobacter fruendi, can produce acid and gas from glucose. All right, any question about the carbohydrate metabolism test? If not, let's move on to the motility test. The motility test uses a semi-solid media, very low agar concentration, so that you have a semi-solid agar, but you stab inoculate. And if the bacteria only grow in the stab, then the bacteria is not motile. But if the bacteria can swim through the semi-solid agar, the bacteria are motile, and they will either appear diffuse like this, where you see there on the side, you can see through it, it's translucent. On here, it's cloudy. The bacteria are all cloudy, and this is a very motile bacteria. Let me get paint up here, and I'll show you what a, got to do that again. For some reason, my computer acts a little different. You got to do it twice to get paint now. So here we have a tube, and we do a stab inoculation. If the bacteria, not very straight stab, if you get a slightly motile bacteria, the bacteria will flare out from the stab. Make this one even bigger. Oops, went too far there. Let me erase that. All right, so if you get flaring, that is a slightly motile bacteria. Any question about any of that? Like I said, it's semi-solid, allowing the bacteria to swim through it because there's so little agar in the motility media. I think it's 0.4%. No, 0.4% agar. All right, so let's talk about the hydrogen sulfide production. You can measure hydrogen sulfide being produced in several different tests. You can measure it in the triple sugar iron test, that's the TSI test. You can measure it in the SEMS test, that's the sulfide indole motility test. Excuse me. And you can measure it in the HE agar plate, or the hectonenteric agar plate. So each of these three different media can look at the hydrogen sulfide being produced. If hydrogen sulfide is produced, the precipitate will turn black. So if you see black in the tube or on the petri dish, then hydrogen sulfide gas is produced. Citrobacter, Salmonella, and Proteus species are all positive for hydrogen sulfide production. Let's look at a positive. This is looks like the Sims tube, and that's a negative control. This one is inoculated with a species that makes hydrogen sulfide. And as you can notice, hydrogen sulfide is usually in the bottom of the tube. Here it's almost the entire tube. It's usually made anaerobically, and that's why the bottom of the tube is black. And the top may not be black because it's not anaerobic. Any questions about hydrogen sulfide production? Let me show you the HE auger plate. Now there's a hydrogen sulfide being produced in TSI tube, also on the bottom. And there's Sims media again, hydrogen sulfide being produced, black. Let me see if I can blow that up. There you can see black colonies on this HE plate. That's because this species is producing hydrogen sulfide. And when grown on the HE plate, you can look for hydrogen sulfide production. All right, any question about any of that? If not, let's move on to the triple sugar iron. You can use this to look for the production of hydrogen sulfide, but you can also look at the growth anaerobically, that's down in the tube, or aerobically on the slant of the tube, and then look at the fermentation of sugars on both the slant and in the bottom of the tube. So triple sugar iron slant test has only 0.1% glucose, but 1% lactose and sucrose. So you notice the lactose and sucrose are 10 times more concentrated than the glucose. It has the phenol red indicator in it, looking for acid production. And then sodium thiosulfite and ferrous sulfite is used to detect hydrogen sulfide production. What you do is on these media, you get your needle, that's that device shown here, which is just a simple wire at a point, it's not a loop, and you take the loop, the needle, and you take it to a bacteria colony, and you can either streak the tip in the colony or you can just roll it in the colony, picking up the cells. And you want to stab inoculate the bottom of the tube. You actually want to go down 2/3 of the way. This isn't quite shown proper. It should go down 2/3 of the way from here, not halfway. So it should go down to about here. You don't want to go all the way down because you don't want to damage the needle on the bottom of the tube. But you do want to go about 2/3 of the way down. You then pull it straight out and then take the tip again, put it in your colony of cells, and then you smear the tip and the cells on the slant in S-like motion or serpentine motion. And that way we'll get growth aerobically grown on the slant and growth anaerobically growth in the bottom portion of the tube. And then compare the growth on the slant to the bottom portion of the tube. All right, in this test tube, we're looking at different results, and it's kind of complicated. If there's no result, then the media won't change in color in the bottom of the tube or the slant. And this would say negative, no sugars are fermented, no gas, hydrogen sulfide is produced, and then no gas is being produced. Here you can see gas being produced both in the cracking of the auger and the lifting of the auger or the bubble. So there's gas being produced in the fermentation. Let's talk a little bit more about that one because this was an easier one to talk about. That's tube 4. Here we have it A, meaning acid is produced on the slant, and A, meaning A over A, acid for the slant, A for acid being produced in the tube. That means fermentation of at least one sugar happened on the slant and in the base of the tube. Glucose will always be fermented because that's the base sugar. And because both are being fermented, we're going to say, or both are yellow, we're going to say plus at least lactose or sucrose, or maybe both of them are being fermented. We don't know if it's one or the other, but we know at least one, lactose or sucrose, or maybe both of them are fermented. And that's because both the slant is yellow and the base is yellow. Gas is produced because it's being lifted, it's cracked. No hydrogen sulfide is being produced because it's not black. Tube 4. So I've explained tube 1 and tube 4. Let's move on to tube 3. Tube 3 is red on the slant, but yellow in the base. Tube 3 only has glucose fermented. How we know that is because the glucose is 10 times less concentrated than the sucrose or the lactose. And if sucrose or lactose were fermented, the slant would be yellow. Because the slant is red, all the glucose in the slant has been used up because the glucose is at low concentration. And so if we had looked at this earlier, you'd normally look at this about 18 hours to 24 hours after inoculating it. And if it's red here, all the glucose was used up. But if you looked at it earlier, that would have been yellow. What happens is the bacteria, once they use up all the glucose, they will then start utilizing protein, which is in the media. Protein, when it gets digested, will make ammonia, which is basic. So initially, this was yellow from the acid being fermented, from the glucose being fermented. But once the glucose gets all used up, the bacteria start utilizing protein, making a base, ammonia, and that will change the color from yellow on the slant to reddish or pinkish. On the base, the sugar is metabolized slower, so not all the glucose has been used up. And it's because of aerobic respiration. The bacteria aren't using aerobic respiration down here because there's not enough oxygen. And so they're using some other means to ferment the sugar, maybe even fermentation. And not all of the sugar is being used up. And that's why it's yellow on the base, but pink here. Glucose is only fermented when it's yellow in the base, but the slant is pink or red. Any question about tube 3? It's also not black, so there's no hydrogen sulfide being produced. Now here, we see that the slant is red. So on tube 2, once again, only glucose is being fermented. And we can't see that the base is yellow, but if the base is black, you can know that the base is yellow. You just can't see it because hydrogen sulfide being produced only happens when the sugar is fermented. In this case, only glucose is being fermented because the slant is red and the base is black, meaning it's also yellow, if you could see the yellow. Any question about tube 3? All right, that's the triple sugar iron test. It is complex. I don't know how you can remember this test, but there it is there. Let's take a look at the SIMS. test. SEM stands for sulfide indole motility test. You're actually running 3 tests at the same time at the SEMS test. In triple sugar iron, what are we testing here? Is the glucose versus glucose and one other sugar. We're looking at the production of gas. We're looking at the production of hydrogen sulfide. And then we're looking at the growth here and here. So it's 3 or 4 different tests being happening at the TSI tube. SIMS has 3 tests. Um, sulfur being produced. If sulfur is produced, you see black. Indole, if indole is produced, you add Kovacs reagent and it will turn pink, red. So all of these are indole positive. This one and that one are indole negative. That one is also in hydrogen sulfide negative. And then we're looking at motility. What you do is you stab inoculate this media, and it's very thin. And if the growth, let me blow this up, if the growth only grows in the stab, then it's not modal. So that one, we only have growth in the stab, so it's non-modal. That one is non-modal. This one has growth away from the stab, so it's non-modal. Here you can't see it, but it's black everywhere, so we can assume that the growth is everywhere, and it's very modal. Any question about any of that? So tube 1, negative in every case. Tube 2, positive hydrogen sulfide production, positive in motility, positive in motility. Motility positive, indole positive, sulfide production positive for tube 2. All right, any question about any of that? All right, let's talk about the McConkey's auger and HE or hectoenteric auger. McConkey is also called MAC. When these 2 tests, we're looking at the growth of cells, and if it grows on the media, it's likely to be gram negative because both McConkey's and hectoenteric agar have ingredients in it that select against gram positive cells. So if there's growth, it's likely to be a gram negative. You can also look for in McConkey's if they look at the lactose fermentation, and if the bacteria cannot ferment lactose, the bacteria will be its normal color, which is not pink. Here, that bacteria is white or off-white. And both the bacteria and the area, the media, the around the bacteria turn pink, and that's because lactose has been fermented, creating an acid, changing the pH, uh, making it look pink. Any question about McConkey's? An HE plate, you have growth, then it is gram negative. No growth on the plate, it is gram positive. If the bacteria can ferment the sugar lactose, it will be a salmon colored, let's say yellow-orange color. If the bacteria cannot ferment the sugar lactose, the bacteria will be its normal color, which is either off-white or white. And then if the bacteria are non-lactose fermenters and it's black, it's also producing hydrogen sulfide gas. Any question about that? Now remember on the MAC and the HE plates, if you have no growth, you cannot assay for the fermentation of the sugar, which for MAC plates is lactose, and for HE plates is also lactose. Salmonella is an organism that is a non-lactose fermenter and produces hydrogen sulfide and will grow on both the MAC plate and the HE plate because it's gram negative. All right, any question about those? If not, let's talk about the lab exercises. You're not physically going to be performing any of these tests, but you are responsible for knowing how to perform and interpret them. We have videos on the carbohydrate fermentation test, on the SIMS test, on the MR-VP test. Remember, that's two tests, the MR test and the VP test. The TSI or triple sugar iron test, and then a video on the McConkey's test. You should be able to complete this lab, meaning uh, the worksheet to this lab module, without going to the site MicroBuzz. The MicroBuzz, I don't believe that website is actually working, but you can use the information in this lab to finish the um, worksheet, and then also complete the case studies, which I've got below. Case study 1, a prison inmate is brought into the ER room with an open wound, and he's known for self-inflicting. A routine culture and sensitivity test is ordered. The appropriate transport media is used for specimen collection and inoculation. And then we have his growth grown on a blood agar plate, and that's showing you some information about hemolytic activity of the bacteria. Let me shrink that down a little. And then we have the bacteria grown on McConkey's plate. It's growing, so that means it's gram negative. And it's pink, so that means something. And then we have the gram stain there, which you have to blow up tremendously to get it so you can see it. Hopefully you can see what it is there. So you should be able to determine the shape and the gram stain from this gram preparation. Any questions about that? You do have to blow it up tremendously to get the shape of the cells. So that gives you some information. And then we have some more information. It's a catalase positive organism and oxidase negative. And day 2, we also did the following tests. Here's the bacteria grown in the SIMS tube. Here's the bacteria grown on the MR tube. Here's the bacteria in the VP tube. There's the bacteria in the citrate test. And there's the bacteria on the triple sugar iron test. Let me show you the worksheet now. So use this key for this lab exercise only, meaning lab module 9, to identify your bacteria, Enterobacteriaceae. Sorry, this tube key was split. And then Pseudomonas here. Case study 1, whether it was oxidase positive or negative, whether it ferments glucose, whether it ferments lactose, what the triple sugar iron results were, H2S results were. This will be positive. This one will be a little more description, and you can either blow this cell up or just write it in. You don't need to fill in this. I'm not going to grade this. A motility, give me the interpretation there. The indole, the MR test, the VP test, and the citrate test. And then from this information, going up to this table, identify the species. And then for case study 1, unknown 1, give me the species right there. Any question about that? And then do the same for case study 2, which I kind of like this one, so I'll read it. Mikey talks his mom into buying a pet turtle. He does well at changing its water, but he's not diligent about washing his hands afterwards. About 2 weeks later, Mikey develops diarrhea, nausea, and fever. His pediatrician orders a fecal specimen and routine culture and testing. Here we have the bacteria grown on a McConkey's plate, and take a look at the colony the arrow's pointing to, and then the colony of interest on an HE plate. So here we're looking at a colony with, there's an arrow there, a small arrow. And that's telling you some information on the HE and the McConkey's plates. It's the bacteria is catalase positive and oxidase negative. Some further tests which were run on day 2. The SIMS tube here, information, the MR tube here, the VP test here, the citrate test there, and the TSI, triple sugar iron test here. So what's the species? And write it in the worksheet there. Also in the worksheet, let me show you the worksheet now. You need to fill in this table for question number 1. What are the positive results of the oxidase test? And I've got the information there. The cells or the spot turn blue. The negative test, pretty easy. It's not blue for the oxidase test. So write in what the positive result is for the indole test, the negative, the MR test, the VP test, the citrate test. All of these you can get from this lab module, maybe not the oxidase test, but all the others. Then the carbohydrate fermentation test you can also get from this lab module. Triple sugar iron you can get from this lab module. You need to tell me what the positive result is for glucose fermentation. And that's to be written in the slant over the butt. And then the negative results. And then the same for the lactose sucrose fermentation, the slant over the butt, and then the negative result slant over the butt. What's the color, the result of the hydrogen sulfide, positive, hydrogen sulfide negative, and then what's the result of gas being produced in the TSI. SIMS tube, give me the results for sulfide production, positive, no sulfide production. Indole being produced, give me the positive result, give me the negative result. And motility, give me the positive result and the negative result. You can abbreviate that, but try and give me the result. The SIMS tube, you can get that information from this lab module. For the growth on MAC and HE, you can also get from this lab module, but give me the results of lactose fermentation and then non-lactose fermentation. And then answer the questions in 2A. 1, what reagent must be added to detect the presence of indole? 2A2, if indole is present, what test will show what color? Going to give me the color. 2B, methyl red is the pH indicator that remains what color under acidic conditions? And methyl red pH indicator is used in what test? Which test uses Barrett's A and Barrett's B position reagents? So that's 2C1 to detect the presence of acetone, a neutral product formed from glucose metabolism. If acetone is present, the test will show what color. So that's 2C2. 2D1, Simmons citrate uses the pH indicator, name the pH indicator, and it turns blue under acidic or basic conditions. So you can bold it acidic or basic, or you can underline or you can type it in right there or write it here. Okay, because that would be 2D2. 2E1, what is the pH indicator used to detect PR carbohydrate fermentation? And it is yellow under either acidic or basic conditions. That's 2E2. 2F, bacteria that possess this cell structure test positive for motility, so name the cell structure. And then 2G, three organisms that produce hydrogen sulfide, name three species. 1, 2, 3. You could name a family, but only if the entire family or genus is positive for hydrogen sulfide production. H, if an organism ferments glucose, but not lactose or sucrose, the TSI slant will be what? And the deep butt will be what color? 2J, state the three tests for which SIMS is used to test, meaning what three tests does SIM measure? 2J1, MAC and HE plates are both what? In that they inhibit the growth of gram-positive, gram-negative cells and of what bacteria? So 2J1, the plates are both what? In that they can inhibit the growth. And then in 2J2, state or underline or highlight, do they inhibit gram-positive or gram-negative? You put that there if you want to write it. And they are also what? In that they separate various bacteria based on visual differences from sugar fermentation. All right, that's it for 2, and then complete the case studies, which I've already talked about. Any question about this lab? If not, I'll be here until probably 8:20 to help you with the lab, so go ahead and get started on the lab. Any questions before I stop recording?